All right. Well, welcome everyone. Thanks for coming to this talk. And I welcome the questions in the middle. If you have, it will be more fun. I will start. So first of all, I'm a faculty in biophysics, and I'm also affiliated with physics. And my lab research is in the field of systems and synthetic biology. So I realize maybe most of you don't or are not familiar with. Again, stop me if you have any questions about any terminology. I'll try to keep it simple. So I'll start by showing you this video here. Sorry. Okay. So this is a huge embryo of Xenopus, and start with one cell that has a diameter about 1 millimeter, so you can see by naked eyes. After fertilization, this huge cell is quickly cleaved into smaller and smaller pieces in a highly synchronized and clock-like manner, about every 25 minutes. And indeed, we know now that this process is controlled by a clock. It's called a mitotic oscillator. This oscillator is formed, first of centered at the cyclin-dependent kinase. It's formed by interactions of a bunch of proteins, which has kinases and phosphatases that can interact with each other to constitute the positive and negative feedback loop. And I'll talk more about the design of the clock. But basically, what you have here is the maternally deposited cyclin B mRNA will translate during the interphase and then bind cyclin B, the CDK1 for activation. And this activation is co-regulated by the double positive and double negative feedback loop. So the switching from off to on is a very sharp manner. And that allows this mitotic entry to be irreversible. So this is about the cell cycle. But cell cycle is just one of the many clocks that control all kinds of cellular and developmental processes. Their time scales can span orders of magnitude. And if you interrupt any clock regulator, you will link this into various diseases. So it's very important for us to understand what maintains the clock function. So what we know here is that even though this clock stay in different forms and they have different molecular mechanisms and also they have very different time scales, but they seem to all share the same function. So first of all, this clock seems to have the ability to adjust their timing in response to different environmental cues, such as light, temperature, or nutrition. And for example, the cell cycle is it's very sensitive to temperature. If for people maybe know some biology, and you know that the E. coli cells can double much faster at a higher temperature. And so on the other hand, the clock must be perform robust oscillations to resist some small perturbations in the environment. So as a physicist by training, one of the central questions that we are interested in is to find out whether we can identify some common design principles that may be shared among all these different clocks to control certain common functions. So that's what I want to also focus on for today's talk. So the question, however, is very challenging to answer, considering that any typical clock network, such as a cell cycle I'm highlighting here, so this is a network we found from the Keck database, and you see that there are hundreds of molecules involved in this network. And additionally, this cell cycle network embedded in the even larger, more complicated networks in any cell. So it would be hard to gain any insight or simple insight out of this complicated network. So what we do instead is to have a approach called the synthetic biology approach. So what we ask is, can we design a minimum oscillator that may be can perform the same function as the more complicated network do, so that we can gain some insights into the fundamental design principles. This question actually dated back to the classical Goodwin oscillator. And in theory, we can build a very simple oscillator that only contains one single negative feedback loop with some sufficient time delay. So here, for example, we can have this little topology here with A as a gene, and it can transcribe into the mRNA as a B node here. And the mRNA can translate into protein C. And if C is a repressor of the gene A itself, that forms a negative feedback. And also, it's important to have these buffer nodes, B and C. So each of these, for example, B for transcription and C for translation, provide sufficient time delay that prevents this single negative feedback into a damped oscillation or damped into a stable steady state. So that's for a theoretical study. And more recently, there is actually engineering of the actual oscillator that is realized by a hallmark study by Michael Alous and Stan Liebler. And what they do is they construct a circuit with 3 genes that repress one another. And this is what they call the repressilator. And they engineered this circuit into the E. coli cells, and they found that about 40% of the E. coli can do the self-sustained oscillations. And 3 years after, Alexander Ninfa's group, who is actually our faculty at Michigan, and they basically construct additional positive feedback that coupled with this negative feedback. And what's interesting about this last topology was that if we can do a survey of all the natural oscillators we know so far, then even though they contain different molecular mechanisms, and again, their periods very different, they seem to all share this topology, which is a coupled positive and negative feedback loop. So the question here is, if we know in theory, a single negative feedback is sufficient to generate limited cycle oscillation, then what is the role of the positive feedback? Why in nature we find these positive feedback structures evolved, and shared among all these different organisms and the different clocks? So this question was first asked in a computational study back in 2008. And in this study, they basically wonder what are the structural modification may change the clock property. So in this case, they're looking at the robustness. So what they do here is they compare 3 topologies, and all of them contain a same core, which is a repressilator that I just introduced. And then in one of these, they add a self-positive feedback and another add a self-negative feedback. And then they compare their performance by a random parameter scan in the parameter space. And if, say, one topology has more parameter sets that's supported for oscillation, then by their definition, this topology is more robust. Okay. So what they found is only the middle one, which is the one with a positive feedback, that seems to have increased the robustness, but not the others. So their conclusion is that the positive feedback can increase the robustness of the clock. But this view seems to be different in a more recent study. This is a experimental work. And what they have here is, again, they synthesized a network, but using E. coli cells. So what you see here, the green E. coli cells are the activator bacteria, and they can not only activate itself, but also activate other bacteria species, which is in yellow. And the yellow bacteria is a repressor bacteria. It can inhibit the activator bacteria through the quorum sensing. So that's the basic structure they first engineered. However, if they engineer additional self-negative feedback loop, they found it's more robust. And so their conclusion here is a negative feedback increase the robustness. So then we see the two studies seems to reach a different conclusion. So what do we think maybe the reason could be that each of these studies only focus on one specific core. However, in nature, we should appreciate that all kinds of designs can achieve the same function, even though some designs may be better than others. But in order to find out the fundamental principles, we have to look at them all. So that actually brings this computational study that I'm introducing now. This is led by a talented graduate student in our lab, Jendali. He's now a postdoc at Stanford. So basically what we look at here is instead of just the one or two core topologies, we look at the whole topology space formed of 3 node networks. And for each topology, we do a random parameter scan. And then we map the result onto this function space. So in this case, we are specifically interested in the robustness of the oscillator. So how do we define the robustness? Similar to the previous study, so what we define here is the number of the hits for within the parameters that we are sampling that can support a certain topology for oscillation. So let's say if in the parameter space, topology 1 has a larger volume than topology 2 for oscillation, then we say topology 1 is more robust. Okay. So with this algorithm, we can identify a total of 1,420 clock topologies. And so what you see here is we arranged all the clock topologies in this atlas here. So each dot here is one topology, and they connect to one another by adding or removing a node or an edge to basically convert to another topology. Okay. And then we also arrange them such that their complexity is increasing from bottom to top. So the bottom is, this axis here is the number of the edges for that topology. So the simplest possible number of the edges you can imagine for a 3-node or 2-node network is 3. And the maximum number of the interactions will be 9, okay? And the fact that all these dots, which is again topologies, can be connected to each other, and there are no isolated dots, meaning that evolutionarily you can always find some optimum path to evolve from a very simple design to a very complicated design. So the first question we wonder is, first of all, we know that this log Q10, the Q value again is representing the robustness. And we see that when we sort robustness from highest to the left and lowest to the right, there are orders of magnitude difference. And then we wonder, because we have the bottom minimum designs as a root for the complicated designs, whether this bottom minimum, we call it cause, can explain the diversity of the robustness for the complicated topologies. Okay. So first of all, what are these bottom cores? So these are actually, so I'll list it here. So this, we call them the minimum designs, because if you remove any node or edge, they just stop have the ability to become an oscillator. Okay. And we also sorted them based on their own Q value. Again, as a reminder, higher of the Q value, more robust this topology is. So we found that the 3 top robust cores have the robustness orders of magnitude higher than the rest of 5. So we call these 3 as robust cores, and then these 5 are non-robust cores. So to answer the question whether these cores can determine the whole atlas, the complicated network robustness, we will group in these topologies. But before that, let's also looking at the robust cores. Interestingly, these three are the ones that we found in nature and in synthetic designs, as I introduced to you before. So namely, these are the Goodwin oscillators, positive plus negative feedback loop, and the repressolator. So now if we group the whole atlas into the groups based on the core composition, and we found that there are a total of 8 clusters, 8 groups. And the very left one are basically containing none of the 3 robust cores, but they can have any combination of the 5 non-robust cores, okay? And then the others are basically having 1 of the robust cores or 2 or 3 of these. All right, so because now we need to compare the robustness across different complexity, we cannot directly use the Q value. Instead, we use the rank percentile for the Q value. So if any topology has a higher, larger number of the rank, is less robust. Okay, keep that in mind. So the very left, the first group, again, it's containing none of the robust cores. And indeed, on average, they are the least robust group that you have comparing to all the others. And also the trend seems to follow what we expect. So basically, the more robust cores you have, the more robust that whole group is. However, this can only explain the average behavior, but it cannot explain the large variation that we also see within the same group. So for example, here, all of the topologies contain the same repressolator core. However, their behavior ranging from very, very high robust to very low robust. So there was something we didn't understand at the time. So I'll keep that in mind. And another thing, the question came up to us was that if we look at the core themselves and just comparing them, so I highlight the core 2 and the core 6 here. So intuitively, I would not be able to tell which one is more robust because both of these topologies contain a slow negative feedback and a fast positive feedback, okay? And however, their behavior differs by a factor of 45. And what's interesting also is that it seems in nature and also in synthetic topology, all of the topologies share the core 2, but none of them seems to have the core 6. So what do we think? Maybe there's something we miss to help us understand this here, was that in addition to the core composition, the local interaction on the node may also matter. So to find it out, we do a pairwise comparison for all the neighboring topologies in the atlas. And for each topology, we decompose them into 2 edge motifs. And the 2 edge motif would be an indication for the interactions on the node. And then we basically calculate what are the difference of the 2 edge motifs and what are the difference of their performance, which in this case R is a rank percentile of the Q value. Okay. So then we use a statistic tool called lasso. So this is a linear regression technique. And we also compare with alternative statistical tools, partial rank correlation coefficient. And we wonder which of these 2 edge motifs plays an important role to increase or decrease the robustness of a topology, okay? And then interestingly, both seems to point to the same clusters. And so what we have at the bottom here are the 3 2 edge motifs that seems to share the same pattern, which is there is a node that receiving different signs of the inputs, and we call them incoherent input interaction. And this seems to increase the robustness of the topology. And the top 4 here also have the shared pattern, so which is a node that have receiving the same signs of the input, and we call them the coherent inputs. And these seems to decrease the robustness. So whether these motifs actually play the role in the topology, we go back to our atlas again. But in this case, instead of group them based on their core composition, we group them simply by counting how many number of the nodes they have the incoherent input and how many numbers of the nodes they have the coherent input, but regardless of the core composition. Okay, so now what you see is that the larger number of the incoherent input and the lower number of the coherent input, the more robust you are for that topology. And indeed, these effects are additive, as you can see from the color map here. So that is interesting because if you want to design a synthetic oscillator with robust behavior, you can certainly have this as an insight to design more incoherent input interactions. And what's also interesting is it seems to explain why core 2 and core 6 have such a different behavior in robustness. And this may be explained by core 2, where both have this self-positive feedback coupled with negative. And the positive feedback is adding to B to generate an incoherent input interaction. But for core 6, it's generating a coherent input interaction. And that may also unify the previous findings. So if you remember at the beginning when I introduced the two studies, they have different findings. So one with a positive, one with a negative that increase robustness. But if we look closer for the node they add for the positive and negative, we found that in this case, they need to add a positive in order to generate incoherent input. And in this case, they need to add a self-negative in order to generate a incoherent input. Okay, so if you can, again, do a survey for all the natural and the synthetic oscillators, you also find these seems to be enriched with incoherent input topologies, as I highlighted the node with green here. Okay, so to conclude for this part, we basically use some computational framework to ask the question whether the topology itself can play the important role for determining a clock function. And the answer is yes. And we also identified the incoherent input interaction that seems to be very important for increased oscillator robustness. And this may explain why in nature we have these peripheral structures that are not really required for oscillation, but they are still evolutionarily well conserved. So they may exist there through the evolution selection by providing some important functions, in this case, robustness. So we now use a similar framework to look at other clock functions, such as the tunability, which I introduce later. But in our lab, when we have this computational study and have some kind of prediction, we also wonder if we can design experiments to test these computational predictions. So what we are choosing here is, again, the cell cycle. So the reason for us to select cell cycle for the study was that the cell cycle is a very typical oscillator that contains both positive and negative feedback loops. And additionally, it has the common clock functions that I introduced at the beginning. So they must be robust against some stochastic noise, either through say cell division, which generate partition errors, or some specific intrinsic noise from gene stochasticity. And additionally, they must be tunable to different cues, temperature, nutrition, etc. Okay, so it's, however, very hard to study in the intact cells. What we are having here in our lab is to develop a artificial cell system by using the microfluidic chamber. So what we do here is we now group all these embryos that I showed at the beginning from Xenopus and fractionize them into these different proportion, different fraction. And then in the middle here is a cytosolic portion where it contains all these molecules such as the proteins and mRNAs that can reconstitute the clock. And now we can inject this, we call it extract, into the microfluidic chamber filled with oil. And that allows us to generate a water in oil emulsion, as you see here. And inside is a extract that can, allow us to reconstitute the cell cycles. And outside is oil. And then it's a cell size, so we can mimic the real cells by using these little droplets. Okay. And then after generating these droplets, we can load and incubate them in the two-dimensional chamber on the microscope. And then we can use imaging processing to segment and track each droplet and then analyze their behavior. Okay. So with this setup, we go ahead and reconstitute the cell cycle circuit. And in addition, we also need to reconstitute a reporter and to allow us to measure this dynamic behavior. So in this case, we have a securin, which is one of the clock gene regulators substrate, and it's an anaphase promoting complex. So what this protein does is it's basically, if we put the mRNA of this thing inside, it translates in the interface, and then dry when the cell enters into mitosis, activate this anaphase promoting complex. It can target a securin for degradation. And then we also attach this protein with a fluorescence reporter, M-cherry. So as you see in the color here, the red indicates the M-cherry intensity. And so I hope you can see on the left here that each droplet, regardless of the size of the droplet, they always have these oscillations from low to high and then back to low again for many cycles. And on the right here, we select one of the droplets for the analysis. So we trace them over the whole time course. And what you see is the oscillations for about 30 to 40 cycles and over multiple days. Okay, so this is probably a simplest possible synthetic oscillator we can constitute about. So there's even no DNA inside. However, if we add a demembraned sperm DNA into this system, it can reconstitute nuclei by forming the nuclear envelope surrounding the DNA. And this is what we call the downstream events that can be controlled by this cell cycle oscillator. And so what's interesting is that this cell cycle seems to be functional to drive the rhythmic dynamics of these two downstream events. One is a chromosome condensation and a decondensation, as you see in the blue here. And simultaneously, this controls nuclear envelope breakdown or nuclear reformation. This is in the green channel. And we use a nuclear localization signal fused with a green fluorescence protein. So what you see here is the NLS, again, it's a signal that always localizes into nuclei. So if there is an intact nuclear envelope, as in the interface, all these molecules will translocate into this nuclei. So you see the bright green dots here. But when the cell enters into mitosis, there is a nuclear envelope breakdown. So all these green fluorescence protein just uniformly distributed in a large droplet. Okay. And again, at the bottom here is securin M-cherry, which is the anaphase substrate as a reporter for the clock. All right, so it may be easier if I just select one of the droplets to look at the correlation of these different events. So at the bottom is a bright field, and here is, again, the cell cycle reporter. And on the top two are the two mitotic events. And I highlighted the mitotic phase. So there are two cycles I'm showing. And I hope you can see at the mitotic phase, there is a correlated events where when the nuclear envelope breaks down, the chromosome condense. Okay. So that's a characteristic of the cell cycle. Okay, so with this system, we want to go ahead and analyze the property of the oscillation. And so the first thing we realized was that regardless of what reporter we have, right? So we have different substrates, cyclin or cyclin B, or different color, the fluorescence reporter, M cherry, CFP, and YFP, they always show this, the profile that has increased the period, increased the baseline, and increased amplitude. This certainly cannot be explained by a standard cell cycle model in the field, which is to predict that you should have a stable limit cycle oscillation that has a stable period and amplitude. So what we think is maybe this system, so it does not have any yolk to provide nutrition. And it's likely that the mitochondria is gone. Mitochondria is an energy factory for the cell to provide energy. So that means each droplet is now has a certain limited energy to begin with. But because this is the out of equilibrium system, over time it should consume energy. So therefore, we should not expect energy as a unlimited reservoir. Instead, we should consider energy into our cell cycle model. So that's what we did. And also, specific design for the cell cycle model was that there is this V1 kinase and CDC25 phosphatase. They are antagonized the regulator for the CDK1 for activation and deactivation. And however, these two also differently regulated by ATP. So if ATP is consumed, you would expect more V1, which is the inhibitor of CDK1, and less CDC25, which is the activator of CDK1. So therefore, the activation of CDK1 will be very sensitive to the energy level. And that simple inclusion of ATP allows us to recapitulate the major property we see experimentally, such as increase the period and amplitude and baseline. Okay, so that actually brings a question to us, which we think is very interesting, is that can energy modulate any oscillation behavior? And that is some question that is seems to be overlooked before. So we wonder if we can design an experimental system to test the idea. So first of all, as a very preliminary experiments, we just add a different amount of the energy mix, which you can equivalent to different ATP you add into the system. And what you find is that within each group, you still have this increased amplitude, baseline, etc. But if you compare across the groups, you find the more energy you add, the less of the period, less of the amplitude, and less of the baseline you have for the system. And if you look closer, by looking at the different phase of this oscillation, so we have the rising phase and the falling phase. And because our reporter here is a cyclin, again, is an anaphase reporter, and we know that the reporter is increasing during the interphase and decreasing at the mitotic phase. So this is also equivalent to that. The rising phase is interphase and the falling phase is mitotic phase. And we found that it seems only the interphase is sensitive to ATP level, but the mitotic phase is very robust to different energy. Okay, and this is only by adding different amount of the energy manually by pipettes, right, by hands. So it's very low throughput and it's not continuous. In order to have a way for tuning the parameter in the parameter space in a more continuous and high throughput way, we are designing a pressure-driven microfluidic device. So here what you see is different channels, each connect to some pressure-controlled device. And so by changing the pressure profile, you can change the fraction of the molecules you want to tune in each droplet. And then we also add the fluorescence dye here to indicate what is the fraction relative to concentration in each droplet for a certain molecule you want to tune. In this case, it's ATP. So what we found is the more ATP you add, And indeed, it's confirming the manual tuning method where you have lowering period in the interface, but the mitotic phase is more or less the same. So, and this is for the simple synthetic cell. But it's also true, seems to be, for the more complicated cell with the nuclei. So here again, we have the NLS fused with GFP. So we use a standard deviation across this, droplet region to indicate for the oscillation behavior. So what we found is, again, over time, the energy seems to be consumed as you see the interface is increasing, but the mitotic phases stay more or less the same. And this can be explained by our cell cycle model, where we can predict a stable mitotic phase and then increase the interface, and likely related to the design of the system with a positive feedback. So you would expect a relaxation type of the oscillation behavior. And indeed, another study in mammalian cell line, they found the positive feedback seems to provide a sufficient insulator to protect the mitotic phase to be changed by the environmental factors. Okay, so now I'm another thing about this project. So now we have the way to do a parameter scan experimentally. We also need a sensor that can specifically measure the ATP consumption in real time. So we have this called a Queen sensor. It can measure the ATP using fluorescence state change. And additionally, we also designed the Fred sensor. And this is also a fluorescence-based sensor. Basically, you have a linker, which is a substrate for a kinase. And the activation of the kinase can change the structure of this linker to bring the CFP and YFP together. And through the energy transfer, you're going to change the fluorescence. Okay, so that's what we measured here using the Fred sensor. All right. Now I'm switching to another project that using this artificial cell system to basically ask, what is the clock tunability designed by? Okay, and this is also another important question because it seems most of the clocks in nature has this ability of the tunability. Actually, in the signaling system, we know that both amplitude and frequency tuning exist. And what people consider was that the frequency tuning seems to be able to coordinatively regulate two genes that have different expression level. And in cell cycle, the same computational study also looking at the tunability in addition to the robustness that I just introduced before. So here what they look at is specifically the cell cycle circuit. And they wonder if the positive feedback strengths can allow the change of the tunability. And based on the computational results, they found that so R here is a positive feedback strength. So they found the higher of the positive feedback, the larger region you can tune in frequency and without changing too much of the amplitude, which is related to the physiological range. So that is what they call the frequency tuning for the clock. So we went ahead using our computational framework to look at a more generalized design. So here I'm showing two examples. So we use the same experimental computational framework for the random parameter scan. But here, with each random scan, we use that as a, that parameter set as a center. And then we explore in one dimension in the parameter space up and down until they reach the bifurcation point where the circuit cannot oscillate anymore. Okay. So what you found is only on the right here, which is a Goodwin oscillator coupled with a negative feedback loop. And we found this topology is very, very hard to tune in frequency. And only if you add a positive feedback, you start to see this frequency tuning mode. So that seems to support the previous finding. And we wonder if experimentally we can test that. So there are different ways of tuning the cell cycle. As I said before, cell cycle is temperature sensitive. So the easier way would be if you can design a temperature gradient, as what we show here, and then looking at, so basically tune the oscillator speed by the temperature. But the problem here is temperature can change not only the cell cycle, but also a lot of things in the cell. So that's not a very ideal tuning method. And instead, we use a more direct tuning because we know this circuit and there is a clock input, which is a cyclin B mRNA. So if we add a different amount of the mRNA, that can change the clock speed. And indeed, that's what we see in the preliminary data. So again, here is a low throughput method. We just add manually different amount of cyclin B mRNA for each group. And then we found there's certain response of the clock, that the clock become faster and faster. So in order to have a high throughput continuous tuning, we again use the channel tuning. And in this case, we use a 3-channel tuning. So for one of the channel, we use cyclin B mRNA to tune the clock speed. And in another channel, we use a drug, and usually it's a small molecule drug, that can target a certain clock design. So in this case, we use a V1 inhibitor that's supposed to link the positive feedback strength. And if we do it simultaneously, we can hopefully explore the clock topology and in relationship with the clock function. So here's our preliminary results. If we add more and more V1 inhibitor, which you can understand as less and less strength of the positive feedback, in theory, we predict is less tuning of the clock. And indeed, that seems to be what you show here, that the range of the period is smaller. And now we explore different clock designs for different candidates of these molecules, trying to understand find out the principles for behind the tunability of the clock. So here, I probably will skip these slides, but basically this is our different projects we can do in the lab using the synthetic cell system. And I want to switch gear to basically show that if you have multiple clocks in an excitable medium, and you would expect some phenomena called the wave propagation. And indeed, this is a widespread in chemical system and physical system. And this is a busy wave, it's a chemical reaction, and also biological system, as you see on the 2 videos on the right here. So this one is actually a xenopus egg with a calcium. And you see that at the very beginning, there is a calcium wave that also behaves as a spiral wave. And this is a fly embryo, so it's a Drosophila. And what you see is the very early syncytial cell nuclear division. And what you see, the target wave from left to right are actually the nuclear division events. And these are the wave with the interaction of the same kind of clocks. But also even in a single cell, we know that there are multiple different clocks, such as circadian clock and cell cycle, and they're not running separately. Indeed, they couple together. And so it would be complicated, but also interesting to study how these clocks couple together to form certain patterns in, say, development and cellular function. So this wave behavior is actually probably the classical one is the action potential that propagating through the axon of a neuron. And we know that some neuron, like motor neuron, can be very, very long. And if just by molecule diffusion, it will be a very inefficient communication. And instead, by action potential, there will be a faster transmission of the signal. And mitotic system seems to share a similar property. And the first mitotic trigger wave was discovered back in 2013. And we are here reconstituting artificial system by basically, instead of encapsulating each nuclei in the droplet, we allow them to share the same cytoplasma. And what you see on the bottom here is called a camel graph. It's just a temporal spatial plot for this tube across. So the y-axis is cross tube length in one dimension. And what you see is these nuclei seems to randomly enter into mitosis at the beginning, likely due to stochasticity. But eventually one clock or nuclei seems to dominate, and that drives this wave that you can clearly see. It's a linear wave. And we can calculate for the wave speed, it's about 120 micron per minute. And we additionally can just reconstitute using cytoplasma without any nuclei. And that also seems to generate this wave, as you see on the left here. What's interesting is that the ones on the left here was we called the chemical wavefront. That seems to propagating faster than the nuclear wavefront statistically. So we still, and this is also true for two-dimensional wave, by the way. And we still don't know what is the mechanism. But one thing we could imagine is that the nuclei can perform as a sink to basically allow the molecules translocated into nuclei to block the communication until nuclear envelope breakdown. Okay. So finally, I think I will also skip this slide, but it's basically just another major direction in the lab using the zebrafish embryo to look at the more complicated patterns that also related to the mechanical signals. And with that, just want to thank everyone in the group. And these are a group of talented postdocs and students. And I want to thank my collaborators and funding support. Yeah, thank you for listening. And if you have any questions, I'm happy to chat.